Welcome to another deep dive. We are so glad you're here with us today.
Absolutely.
For this deep dive, we are zeroing in on a single, just incredibly comprehensive source. It's a 2023 scientific review article published in the journal Biology.
Right. Authored by Utami, Maxim, and Dayawardi.
Exactly. And our mission today is really fascinating. We are looking at a traditional medicine with a 400-year history. It's a natural plant compound called berberine.
Yeah.
And we're going to explore exactly how it's holding up to rigorous, modern biochemical analysis as a serious treatment for diabetes.
And I think we need to say up front, the stakes underlying this research really could not be higher. When we look at the data laid out in this paper, the scale of the global diabetes epidemic is almost difficult to fully comprehend.
It really is staggering.
It is. In 2021, the International Diabetes Federation reported that 537 million adults globally were diagnosed with diabetes.
Wow, over 500 million.
Over 500 million. And the projections anticipate that number hitting 783 million by 2045.
That's just wow.
We're currently looking at a disease that causes up to 6.7 million deaths every single year. It's a massive, compounding global health crisis that really requires a multifaceted approach.
And even if you listening right now do not have diabetes, understanding how our bodies manage energy, sugar, and fat at a cellular level is basically the ultimate hack for overall metabolic health.
Exactly.
Talking about the core machinery of how your body processes the food you eat and turns it into the energy you use to live. So to understand how berberine fits into this picture, we first need to look at the limits of our modern medical paradigm.
Right, we have to set the stage.
Assuming we all understand the baseline difference between an absolute lack of insulin type 1 and the insulin resistance that defines type 2, the source, really gets into the weeds of what drives that resistance. It's often driven by lifestyle, obesity, and the accumulation of specific fatty acids in the body.
Things called ceramides and diacylglycerol.
Right, which essentially gums up the cellular machinery.
Yeah, those waxy lipid molecules physically interfere with the signaling pathways. The cells are effectively ignoring the insulin signal knocking at the door, basically because the pathway is blocked.
But the source highlights a fascinating subtype of diabetes that is rarely discussed in mainstream medicine.
Mitochondrial diabetes.
Yes. I found this part particularly surprising because, we are so conditioned to look straight at the pancreas or at dietary sugar, not at the mitochondria when we talk about the mechanics of diabetes.
It's a critical, often overlooked piece of the puzzle. Mitochondria are the powerhouses of the cell. They produce ATP, which is our fundamental cellular energy currency.
Right.
But mitochondrial DNA has a very high mutation rate because, unlike the DNA in the nucleus of our cells, it lacks a robust DNA repair mechanism. The paper points to specific mutations, such as the A3243G mutation.
Okay.
When this genetic error occurs, the mitochondria simply fail to produce enough ATP.
Wait, so if the cell is operating on a severe energy deficit, isn't that going to compromise any system that requires a lot of mechanical effort?
Exactly. That energy deficit is exactly what breaks the insulin secretion mechanism. The pancreas is an incredibly energy-intensive organ. Pumping out insulin requires a massive amount of ATP.
That makes sense.
Yeah, without sufficient energy from the mitochondria, that pancreas just loses its mechanical ability to secrete insulin properly.
Which is a totally different mechanical failure than standard insulin resistance driven by fatty acids.
Right.
And it really highlights why standard one-size-fits-all treatments might not be the silver bullet we want them to be. Speaking of which, the current synthetic drugs we use for type 2 diabetes medications, like metformin, rosiglitazone, and sulfonylureas.
They come with some fairly heavy baggage.
They really do.
The paper explicitly outlines the toxicity levels and the complex side effect profiles of these current first slugs. We're talking about severe gastrointestinal issues and a very high risk of significant weight gain.
Which seems completely counterproductive for metabolic disease.
It is. Plus, drugs like metformin and rosiglitazone are contraindicated for patients who already have liver issues, kidney impairment, heart failure, or respiratory problems.
And even if a patient can tolerate them without those severe side effects, they aren't always fixing the broader metabolic disaster happening in the body.
No, they aren't.
The clinical trials mentioned in the source show that metformin is often insufficient to control the dangerous comorbidities of diabetes. For example, 33.8% of patients on metformin still show persistent hypertension.
That's a huge percentage.
Another 16.6% still show microalbuminuria, which is a protein leak in the urine signaling ongoing kidney damage.
Right.
Okay, let's unpack this. If our modern synthetic drugs are potentially toxic, can cause weight gain, and fail to control all the downstream organ damage, we clearly need to look for alternatives.
Which brings us to berberine.
Exactly.
Berberine is a natural compound, specifically classified as an isoquinoline alkaloid. It's isolated from the roots, stems, and bark of plants, notably from the genus Berberis and others like Corydalis tercinovi.
Try saying that 3 times fast.
I'd rather not. But as we mentioned at the top, it has been a staple in Chinese, Indian, and Middle Eastern traditional medicine for over 400 years. It's often used for digestive issues and infections. What modern science is doing now is applying cutting-edge biochemistry to break down exactly how it works at a molecular level.
And that brings us to the structure-activity relationship, or SAR.
Yes, crucial concept.
It's a concept in pharmacology where scientists look at the actual geometric shape of a molecule to figure out why it behaves a certain way in the body. The paper compares berberine to other similar alkaloids found in these traditional plants, like copticine, palmitine, and epiberberine.
Molecular shape truly determines a compound's destiny in the body. When researchers analyzed these various compounds, they found that berberine and copticine are highly effective at combating high blood sugar and lowering dangerous lipid levels.
And the reason comes down to a specific structural feature, right?
Exactly. A unique arrangement of oxygen and carbon atoms at key geometric points on the molecule.
The researchers found that if you swap those specific parts out for different chemical groups, like what you see in some of those other plant compounds, the molecule completely loses its power to lower blood sugar.
It ceases to function as an antidiabetic agent entirely. However, the paper points out another critical detail about structural modification.
Oh, right, the oxidized version.
Yes, epiberberine. It possesses an oxidized form of that specific chemical group, and it performs an entirely different but equally vital function. It is incredibly effective at inhibiting an enzyme called aldose reductase.
Let's talk about why inhibiting that enzyme is so important, because the source goes into detail about the damage it can cause. When a patient is in a state of hyperglycemia, there is simply too much raw glucose floating in the blood.
Right.
Aldose reductase is the enzyme that starts turning that excess, unused glucose into a sugar alcohol called sorbitol.
And that conversion process is highly destructive. Creating that sorbitol rapidly depletes the body's natural antioxidant stores. Without those antioxidants, there's a massive accumulation of reactive oxygen species, unstable molecules that physically damage cellular structures.
It's like a rust spreading through the body.
That's a great way to put it. This specific sorbitol cascade is the primary driver behind the severe complications of diabetes. The degradation of heart tissue, the stiffening of blood vessels, retinal damage in the eyes, and the destruction of the kidneys.
So by inhibiting aldose reductase, that oxidized structure in epiberberine essentially stops that toxic cascade before it even has a chance to start.
Exactly.
It's like finding the exact right key for a very specific, very dangerous lock. But let's look at how standard berberine actually works inside the cell to lower the initial blood sugar.
Let's do it.
To understand this, I like to think of your cells like a smartphone. When your battery drops to 10%, your phone automatically goes into low power mode. It dims the screen, it pauses all the background apps refreshing, and it forces the entire operating system to conserve energy and optimize its essential performance to survive. In your cells, that low power mode switch is an energy sensing signaling system called AMPK.
What's fascinating here is how berberine specifically targets the mitochondria to flip that switch. Berberine intentionally inhibits complex I of the mitochondrial electron transport chain.
Interestingly, this is a very similar mechanism to how the synthetic drug metformin operates.
Yes, it is. By inhibiting complex I, berberine intentionally but slightly suppresses the mitochondria's ability to biosynthesize ATP.
So, by artificially lowering the production of energy, it's shifting the ratio of molecules in the cell. If ATP goes down, the ratio of the lower energy molecule, AMP, goes up.
The cell senses this shift, thinks it's suddenly starving for energy, and flips on that AMPK master switch to survive.
That artificial energy panic triggers an incredible domino effect to restore balance, doesn't it?
It really does. First, activating AMPK triggers a complex chain called the ACT signaling pathway. This pathway sends a biological directive that tells the cell to take its GLUT4 transporters, which are essentially internal doorways used for absorbing sugar,
Right.
and migrate them right up to the plasma membrane of the cell.
The cell essentially opens all its doors and starts furiously sucking glucose out of the blood to get more immediate energy. It forces glucose uptake even in cells that were previously locked down and insulin resistant.
And it doesn't stop there.
Perfect.
Berberine also suppresses a process called gluconeogenesis.
Gluconeogenesis is the mechanism by which your liver creates brand new glucose from non-carbohydrate sources.
Right. For a diabetic patient who already has too much sugar in their blood, the liver pumping out even more glucose is the absolute last thing they need.
Definitely.
Berberine intervenes by inhibiting a specific genetic transcription factor in the liver. This effectively stops the expression of the key enzymes the liver utilizes to manufacture that new glucose.
So it forces the muscle and fat cells to eat the sugar already circulating in the blood and simultaneously tells the liver to stop making more.
It is a highly coordinated mechanism.
Here's where it gets really interesting. Berberine also actively increases the secretion of insulin from the pancreas, and it does this through a profoundly unexpected biological pathway.
The bitter taste pathway.
Yes. The source explains that berberine boosts levels of GLP-1, a crucial hormone for insulin release. And it triggers this release by activating a cellular receptor known as TAS2R. That receptor is the body's sensory mechanism for bitter taste.
It's a remarkable evolutionary pathway. GLP-1 is an incretin hormone, meaning it's released by the gut in response to food. When those bitter taste receptors in the intestines are activated by the berberine compound, GLP-1 levels rise.
And that kicks off another chain reaction.
Exactly. This activates a specific enzyme that ultimately causes a massive spike in intracellular calcium concentration inside the pancreatic beta cells.
That sudden rush of calcium is the physical trigger that prompts the pancreas to migrate and release its stored insulin granules directly into the bloodstream. It's just fascinating that a 400-year-old bitter herb is essentially tasted by the gut to trigger an insulin dump.
And berberine doesn't just deal with sugar, it goes aggressively after fat, too.
Yes, the paper details how it acts as an anti-adipogenic or anti-fat compound.
This is a crucial function because, as we established at the beginning of our deep dive, the accumulation of fatty tissue is a primary driver of the insulin resistance itself.
So how did they study this?
The researchers utilized an in silico study, which is a highly advanced computer simulation that models millions of potential molecular interactions to observe molecular docking. They found that berberine binds directly to PPRG.
PPRG.
Yeah, it's the main genetic regulator that acts as the control room for adipogenesis, the creation of new fat cells.
And the simulation showed that berberine binds to this regulator with an energy of negative 8.4 kilocalories per mole. To put that in perspective, the body's own native ligand. The molecule that normally binds to this receptor to trigger fat creation only binds with an energy of negative 7.6 kilocalories per mole.
Because berberine has a stronger, more favorable geometric fit, it outcompetes the body's natural signals and effectively shuts down the expression of fat-making enzymes.
That is incredible.
If we connect this to the bigger picture, stopping that initial fat accumulation is vital for stopping systemic inflammation. Excess visceral fat doesn't just sit there inertly storing calories.
Right, it's not just a storage tank.
Not at all. It functions as an active endocrine organ, secreting pro-inflammatory cytokines, things like TNF-alpha and interleukin-6. These inflammatory signals travel through the portal vein and physically disrupt the insulin signaling pathways in the cells throughout the body.
Imagine the cell's insulin receptors are like satellite dishes, waiting for a clear signal from the pancreas. That excess visceral fat acts like a massive radio jammer, pumping out these inflammatory cytokines that completely blocks the insulin signal from getting through.
Which directly causes the resistance.
Exactly. Which means berberine has to act like a cellular fire extinguisher to clear the static.
And the source details exactly how it achieves this. Berberine blocks a specific genetic transcription factor that is normally responsible for pumping out those inflammatory cytokines. It accomplishes this by keeping the factor's natural inhibitor protein perfectly stable.
Because that inhibitor stays stable and locked in place, it masks the genetic signals and keeps the entire inflammation engine turned off.
And that fire extinguisher effect extends to oxidative stress as well. We mentioned earlier that berberine intentionally slows down complex I in the mitochondria to trigger that energy panic. Well, that same mitochondrial complex is also a major contributor to the daily production of destructive superoxide free radicals. By slowing the complex down, berberine cuts off the production of these damaging molecules right at the source.
Furthermore, it triggers a separate cellular pathway that boosts the body's own natural antioxidant defenses. It upregulates the production of enzymes like superoxide dismutase and glutathione, which are the body's internal sweepers that neutralize oxidative damage before it can harm the tissues.
So it's stopping the production of the bad stuff and simultaneously boosting the production of the good stuff.
It's defending against the exact oxidative stress that leads to those horrific diabetic complications we talked about earlier. But I know what you're thinking. This all sounds incredible in a highly controlled computer simulation or an isolated petri dish, but what happens when humans actually take it?
That's the real test.
We have clinical trial data for that. The paper cites a study involving 116 people diagnosed with diabetes. They were given 1 gram of berberine a day for 3 months. The results showed a 20% drop in fasting blood sugar and a 12% drop in HbA1c.
Which is the vital metric that measures a person's aggregated average blood sugar over a 3-month period.
Exactly. And on top of that, they lost an average of 5 pounds, specifically dropping up to 3.6% of their body fat.
Those are highly significant clinical results, entirely comparable to the efficacy of first-line oral antidiabetic drugs. But what separates berberine from those synthetic drugs is its vastly superior safety profile.
Just a huge.
The acute toxicity tests conducted on animal models found that the estimated safe oral dose for humans is exceptionally high, 2.97 grams per kilogram of body weight. In the human clinical trials, the side effects observed were limited to minor gastrointestinal issues, things like mild constipation or flatulence.
So it entirely avoided the massive weight gain and severe systemic risks associated with drugs like rosiglitazone. And this is but there is a major plot twist in this story.
There always is in pharmacology.
Right. Despite these amazing cellular mechanisms and strong clinical results, standard berberine has a glaring flaw. Its oral bioavailability is remarkably terrible. The paper notes that less than 1% of the berberine you swallow is actually absorbed by your intestines into your bloodstream. About half of it isn't absorbed by the body at all.
It's a profound hurdle. You can have the most mechanically effective molecule in the world, one that fits perfectly into the cellular receptors, but if the human digestive tract cannot absorb it and deliver it to the bloodstream, its therapeutic potential is severely bottlenecked.
However, the source provides a fascinating biochemical solution to this exact problem, a natural derivative called oxyberberine.
This is where it gets brilliant.
When that standard berberine hits the intestines and fails to be absorbed, it doesn't just sit there. It interacts with our gut microflora, the trillions of bacteria living in our digestive tract.
Those specific bacteria actively metabolize and oxidize the berberine, transforming its chemical structure into oxyberberine.
And oxyberberine fundamentally changes the equation.
It does. It is highly absorbable and has an incredibly high safety profile. In the required animal safety models, the lethal dose was found to be over 5,000 milligrams per kilogram, making it vastly safer than even standard berberine.
And even more importantly, extensive studies found that this oxidized, bacteria-created form is actually superior to metformin in its antidiabetic effects, and it exhibits even stronger anti-inflammatory properties than the original compound.
That's the key.
So what does this all mean? It means we are looking at a metabolic multi-tool that leverages a 400-year history to fix systemic dysregulation at the deepest molecular level.
It really hits everything.
It forces cells to open their doors and uptake glucose. It tells the liver to stop manufacturing new sugar. It outcompetes our own biology to block the genetic drivers of fat creation. And it shuts down the inflammatory static that causes insulin resistance in the first place.
It truly reminds us that nature holds incredibly complex chemical keys. But it requires the rigorous application of modern science, from simulating molecular docking on supercomputers to developing a deep understanding of our own gut microflora, to unlock their full potential and overcome physiological hurdles like poor bioavailability.
It is the perfect marriage of ancient wisdom and modern biochemistry. And it leaves you with a really profound thought to mull over. Think about this. If berberine's true, highly absorbable, superpowered form, oxyberberine, is only created after it interacts with the specific bacteria living in our intestines, how many other ancient traditional medicines are actually relying on the hidden chemistry of our gut microbiome to do their healing?
That is a fascinating question.
Could the actual secret to ancient medicine not just be the plants themselves, but the bacteria living in our stomachs reacting to them? Something to explore and think about. Thank you so much for joining us on this deep dive, and we will catch you on the next one.